Hello everyone, in this video we would be looking at chromatography. Chromatography is a well-known method that is used to separate compounds present in a particular sample. This method was first discovered by Michael Sievert in the year 1900. Michael Sievert used plant sample and he separated several pigments from this plant sample. What he did was that he used a glass column, loaded the glass column with calcium carbonate, and then he placed the plant sample on top of the calcium carbonate. The brown color that you see here is the plant sample. Then he introduced solvent or liquid that helps in separating all the different compounds present in this sample. Now as he adds the liquid, the compounds get separated in the form of different colored bands. Because of all these different colors, Michael Sievert termed the name chromatography for this particular technique, where chroma means color and graphing means writing. There are several components that are associated with chromatography, and the first important component is the solute or the analyte. So these are nothing but these are the samples that are used for analysis. For example, plant sample, as I discussed in the previous slide, that is one of the analyte or solute. Then we have the second important component, that is the solvent. These solvents are nothing but these are chemicals that are used to dissolve the solutes, and solvents are of different kinds. The different types of solvents could be because of their polarity. Now what is this polarity? Polarity is nothing but it is a charge of a particular compound or a particular molecule. Several molecules have uneven distribution of electron, and because of this uneven electron distribution, these molecules can either have positive charge, neutral charge or negative charge. Now all of these charged substances, we call them as polar, or we term this particular charge concept as polarity. Now polar solvents have high polarity or they are high in charge. For example, water, acetone and ethanol. And then there are other set of solvents such as non-polar solvents which have low polarity. For example, hexane, benzene and toluene. There are also certain solvents that are intermediate polar, where the polarity is moderate. For example, chloroform and diethyl ether. Now this is how, based on the polarity, the solvents are categorized as polar, non-polar and intermediate polar solvents. But one important thing is that not only the solvents, solutes or any compounds for that matter can be categorized into polar, non-polar and intermediate. So one important thing to note is that like dissolves like. For example, polar solvents can only solubilize polar substances. For example, water is a polar solvent and this water can only solubilize polar solutes like sugar, salt, etc. So that is how like dissolves like. Now one important thing to note, to remember, is that polar solvents and water-based solvents are similar, which means water is a very good example of polar solvent. And here this is a very good cartoon that will help us remember that polar equals water. Here there is a polar bear crying for help, saying that I am dissolving, someone please help me. And then another bear comes and this bear tells that, see, bears are generally insoluble. How are you telling that you are solubilizing or dissolving? Now this polar bear says that, see, it is easy for you to say because you are insoluble, you are not polar, whereas in my case I am polar and I am dissolving in water. So that is how you can remember that polar solvents are water based solvents based on this cartoon. There are other phases of chromatography or compounds of chromatography, including stationary phase and mobile phase. The stationary phase are nothing but these are solids or liquids that help in binding the analyte or the sample. For example, solids could be paper or any sheet that helps in binding of the sample. Here this is an example of paper chromatography. Here you can see there is a paper that acts as a stationary phase. The stationary phase are generally non-moving, which means they are stable and they help in binding of the analyte. Here the paper is non-moving, whereas the sample that is spotted is bound to the paper. So this is how stationary phase are non-moving phase and they help in binding of the analyte. Secondly, we also have mobile phase, which is the moving phase as the term indicates. This phase could be either liquid or gas that moves through the stationary phase. In this example, the mobile phase is a solvent or a liquid that passes through the stationary phase. As you can see, this is a sample spot placed on the stationary phase, and as the mobile phase passes through the sample spot, we can see that the sample spot is differentiated, it gets separated into different bands, and this is how the mobile phase moves through the sample. Now it is easy for us to know that when we look at stationary phase, it is easy to say that solids are stationary, but how can liquid be a stationary phase? So I'll give you an example for this. Let's say you have a paper and a sand. Now we know that papers are stationary phase and sand, consider sand to be a sample or analyte. Now when you sprinkle sand onto the paper, do you think the sand will stick onto the paper? No, right? Because there's nothing to bind the sand onto the paper. Whereas in another case, let's say you have a paper that is already wet. Now in this case, if you sprinkle sand onto the paper, obviously the sand will stick onto the paper because of the liquid or the moisture that is present on the paper. So this is how liquid can also help in binding the analyte to the stationary phase, and that is how solid and liquid both can act as stationary phase. So these are the four important components of the chromatography, that is the analyte or the solute, then the solvent, stationary phase and the mobile phase. Coming to the applications of chromatography, so far we came to know that one important thing that chromatography does is that it helps in identifying different components of a sample, and it also helps in separating different components of the sample. So based on that, chromatography is used in wide variety of applications. So the first one is pharmaceuticals, wherein chromatography is used for drug purification, to check the quality of the drug if there are any adulterants or contaminants, to perform metabolite analysis, which means let's say you intake a particular drug and the drug gets metabolized, right? What if the resultant of the metabolite, all the metabolite products, what if some of the products are toxic to you? So all of that analysis can be done by using chromatography. Secondly, we have environment. In environment, chromatography is used for testing the water, air quality, soil quality. It is also used to trace if there are any minor or trace elements in the soil sample. For example, toxins, heavy metals, all this can be done. Thirdly, we have food and beverage. For food and beverage also, chromatography is used for food safety, to detect if there are any adulterants, any toxins, poison, etc. It is also used to analyze the flavor or fragrance of a particular food, and as I said, adulteration can also be identified by using chromatography. Fourthly, we have cosmetics, where for ingredient testing, fragrance analysis and for stability testing, chromatography can be used. In clinical diagnosis, especially in diagnosing diseases, chromatography plays a very important role. It is also used in developing drugs and constantly monitoring drugs in the body fluids like blood, urine. When we take all of these samples, we can detect the amount of drug or any toxin that is present in the particular biological fluid using chromatography. Chromatography is also used for genetic and proteomic studies, where we can either identify genes, proteins, nucleic acids, or we can also isolate them using chromatography. Then in chemical industry, it is also used for component analysis and to check the quality of the chemical that is being produced. In forensic, chromatography is used in detecting the drugs, detecting if there are any drugs, alcohol, poison that are present in the sample, or if there are any explosive, the cause of death or killing can be identified by using chromatography. And in biotechnology, especially in research field, chromatography is used to purify proteins, DNA, RNA. It is also used to study enzyme, etc. So these are the important points that one should understand in the concept of chromatography. So in this video we looked at what is chromatography, what are the different components and its application. In the next video we would be looking at the different types of chromatography.